The living world, evolution of biodiversity. This unit describes biodiversity of Earth and how it came to be, and how environmental factors can cause and influence its decline. So biodiversity can be described as at a genetic level, at a species level, and then at an ecosystem level. The living world on biodiversity, speciation, and the pace of evolution. Speciation works at the process or at the pace of as fast as evolution can occur. So by the end of this, you should be able to explain the process of allopatric and sympatric speciation and understand that the factors that affect the pace of evolution can be numerous and diverse. When I say that speciation can be allopatric or sympatric, it means that new species commonly evolve through these two specific processes. Um, the process of speciation that occurs in like a geographic isolation is allopatric. And when evolution of one species into two, like they split, without there being any sort of like geographical isolation, we call that sympatric. One common way that evolution creates a new species through geographic isolation, basically that means that there's been some sort of physical separation of a group of individuals that are or were once the one species. It's called allopatric speciation. And this geographic isolation can occur when a subset of individuals from one larger population colonizes a new area, a new habitat, and now they've been physically separated from their original population. This leads to reproductive isolation, and as a result, the two populations now are completely separate species. To the point where they can no longer interbreed and produce viable offspring. So here I have an example of allopatric speciation in mice. So there's an original field population of mice, and then a river comes through and basically divides the one set into two different populations. And so they don't swim. So over many generations, this population has become independently evolving, and you can see in the illustration they are showing like there's color change. So eventually, the two populations become so genetically different and distinct from one another that they've spent all this time by themselves, and now they've created a separate species. So even though the river dries up and the two populations kind of come back together, they're no longer going to interbreed because they don't look alike, and they may actually no longer physically be genetically capable of reproduction. Allopatric speciation is thought to have been the most common way in which evolution generates like a new species. However, it's not the only way. Sympatric speciation is the evolution of one species into two groups without there being any sort of geographic isolation. It usually happens in a process that's called polyploidy. Most organisms are diploid. They have two sets of chromosomes. And in polyploidy, the number of chromosomes increases to like 3 or 4 or 5 sets. This increase can occur during the division during reproductive cells, during meiosis, and it's either accidentally or in nature. It can be a result of humans deliberately doing it. Plant breeders do this a lot. They've found lots of ways to create a polyploidy, more DNA in certain vegetables, and like strawberries have like 400 chromosome sets or something ridiculous like that. But polyploidy organisms can also be animals like snails, salamanders, and polyploidy often results in plants that grow larger fruits. In animals, however, it can be a feature that organisms that were once all genetically diploid have now become polyploid, and that generally cannot interbreed with their diploid population anymore. And so as a result, there is this instantaneous separation amongst one species, and they become reproductively isolated. Darwin's finches were believed to be a result of allopatric speciation, where all of these finches had evolved these certain mouth parts just based on like where they were at. We know that we have a common ancestor, but based on the type of food that they were able to eat, these guys were all on the exact same island, but there was a space that was created, and so they were reproductively isolated based on their food source. There's lots of examples of this, and the pace at which this happens can be affected by multiple different ways. So like the finches, Sympatric speciation of flowering plants like wheat, they commonly form new species through a process of polyploidy. And so this has increased the number of sets of chromosomes beyond its like 2 normal sets. And when you increase the number of sets of chromosome, you increase its protein synthesis process, it produces more tissue, and in wheat it actually grows larger. And so wheat used to be this tiny little seed plant, and over time breeding these to become polyploidy, producing more and more sets of chromosomes, we now have common wheat, which is much bigger. And I think this type of wheat has 6 or more sets of chromosomes, and so they're producing these really large seed pods. And so with larger seed pods, larger tissue, that equals more food. As a linologist, my favorite example of evolution at its own pace is what we call rapid evolution. And rapid evolution occurs it's a really significant change over a species genotype and its phenotype. And it's when it has these adaptations that are evolved to find a completely different food source. And it can take anywhere from hundreds to millions of years for this to happen in any species. So let's consider a couple of different examples of rapid evolution. In this illustration, you can see cichlids. So in Africa, in the African Great Lakes, exists a population of cichlids. And as you can see, there's a lot of variety. So evolutionary speaking, evidence that we have indicates that there's roughly about 200 different species of these cichlids in the lake that have evolved from just one ancestor over millions of years. So during this time where this evolution was occurring, some of these cichlids specialized to become eaters of one particular thing, like insect eaters. Some of them became fish eaters. Some of them evolved to eat like invertebrates, like snails and clams. And so although that the cichlids of in Africa have evolved quickly in terms of like evolutionary terms, lots of organisms like the pufferfish of Death Valley, California, have evolved even more rapidly, like in 20 to 30,000 years. I've seen lots of these fish before. They're very popular aquarium fish. They do all kinds of very specific things. Like there's one of these guys that only eats the eyeballs of other fish. There are certain kinds that nest. They make nests inside of the rocks. There's some that live in an under vegetation that's in the water. There's an open water kind, but they all have this one common ancestor, which is why when you just look at them, they look like they're the same fish, but they're just different colors. But they're separated by how they eat and the types of things the food source that they've gone after. So the pace of evolution depends on lots of factors, and it can be very rapid and fast or not. A species can survive an environmental change if it can quickly recover and evolve adaptations to these new conditions in their environment. Slow rates of evolution occur when a population has like long generation times, like elephants that can, they carry their young and they're pregnant for 18 months. I remember one biologist telling me that the African bush elephants, they're the ones with the big, beautiful, puffy ears, they're pregnant as long as 22 months. There's just a lot of animals that have this very long gestation period, and so evolution between them is very slow. Sometimes there are species that their reproductive cycle revolves around the moon phases, and so it's hit or miss, and so that can take a long time too. But evolution by artificial selection, like we saw in the wheat, can be very rapid. So genetically modified organisms, also known as GMOs, those are organisms that are produced by copying genes from one species that we like, that's desirable, and inserting it into another. And so that occurs in a lab and very quickly. A lot of examples come to mind are our foods, like the orange. We have cut genes from one type of species and put it into the orange that you eat in the grocery store to form these very, very thick skins. We basically like beefed up their immune system to insects so that when you get the oranges, they're beautiful, right? They haven't been destroyed by insects. And most of our produce this has happened to most of our produce on a huge scale, to where we have lost some of the original DNA and genes from their original species. Scientists have found that this soil bacteria, it's called bacillus, which naturally produces an insecticide as a defense of being consumed by insects that live in the soil. Plant breeders have identified this bacteria and also the gene inside it that's responsible for making this insecticide. They've copied that gene and inserted it into crop plants like vegetables and such. And so these crops that can now naturally produce their own insecticide, which makes them less attractive to insects, are now common. So we call them like BT corn and BT cotton, and they're named because of this bactrum that was found in the soil. And so, as you might guess, inserting these genes into another organism is a much faster way of producing a desired trait than a traditional plant and definitely a traditional animal breeding system. And so now this natural available variation in the population has changed rapidly.